Hello, my name is Jessie Blumenfield, and I'm a fourth-year neuroscience graduate student at UCSF. I'm currently working in the lab at Yadang, and I study Alzheimer's disease. Alzheimer's disease is a progressive neurodegenerative disorder characterized by a severe decline in memory and cognition. Pathologically, this disease is defined by the presence of amyloid beta plaques and neurofibrillary tangles containing hyperphosphorylated tau, as well as brain atrophy and inflammation of the brain's resident immune cells. Though this disease has been studied for over a century, the biology driving Alzheimer's remains unclear. Interestingly, however, there is one gene, known as the apolipoprotein E, or ApoE gene, that has been found to play a critical role in determining Alzheimer's fate. There are three main alleles of this ApoE gene. The most common one is known as ApoE3. However, there is also one known as ApoE4, which has actually been identified as the single greatest genetic risk factor for late-onset Alzheimer's. This is because carrying one copy of this ApoE4 allele confers about a threefold greater risk of developing Alzheimer's, while carrying two copies increases this risk to 12-fold. On the other side of things, carrying a copy of the protective ApoE variant, ApoE2, actually decreases the risk of getting Alzheimer's by half. In addition to these three main variants, it's worth noting that there have also been rare ApoE mutations recently discovered that actually seem to decrease disease risk as well. Despite this clear role that ApoE has in determining Alzheimer's fate, the mechanisms behind how this protein regulates downstream pathologies is still not entirely understood. Leveraging interstitial brain fluids collected from both disease-destined and disease-resilient ApoE models of Alzheimer's disease, we aim to characterize the factors downstream of ApoE that are disease-inducing or disease-protecting. To do this, we are using microdialysis. Microdialysis is a special dialysis technique which enables us to sample the proteins and other factors being actively secreted in the brains of freely moving mice. Specifically, we perform this dialysis in the hippocampus, which is the memory and aging center of the brain and the most vulnerable region in Alzheimer's disease. Using targeted assays as well as unbiased proteomic techniques to examine these interstitial fluid samples collected using microdialysis on disease-destined and disease-resilient ApoE-type mice, we aim to uncover the key cellular pathways that are important for triggering the cascade of AD-related pathologies. We hope to use this information to identify factors that may provide a useful therapeutic target. Thank you.